Hello, everyone. My name is Chen-Hsiang Yeang, and here I'll just give a very quick introduction about this poster. So in this work, we are building an association structure between the alterations on DNA's, genomic alterations, and variations on transcriptomes or mRNAs. And we call this hierarchical association structure, or IHAS. And we apply this as a framework to TCGA data and identify a very rich association structures. So this slide gives an overview about the IHAS framework. First, we build the associations in a hierarchy from models, modules, supermodule group, gene group, and the metagene groups and the sample groups, as well as the pan-cancer sample groups. And this hierarchical structures is first aligned with the cancer type specific phenotypes, and then aligned with the pan-cancer characteristics in the TCGA data. And finally, we validate the IHAS structures in over 300 external data sets, and many of them are consistent with our predictions according to the TCGA inference results. And this slide gives an overview about the integrated information you can extract by applying IHAS to each type of a cancer. For instance, this is the breast cancer data. And for the associations, we have the targets, which are the MR expressions, and the factors, which are the alterations on the DNA, such as the mutations or the copy number variations. So here on the left panel, you can see the enrichment of each supermodule, functional enrichment of each supermodule. In the middle panel, you see the recurrent factors in each supermodule. On the right, we have the combinatorial expressions of the supermodules across the sample groups. And we also put the PAN50 subtypes on the top and information about survival times on the further. So this gives an overview about one specific cancer type, and we can also integrate those information from multiple cancer types together. So here it shows the combinatorial relations of the supermodule groups, and the gene groups. So we constructed these 217 supermodules and clustered them together to form 17 supermodule groups. And those are the clusters of the columns. And each supermodule group is enriched with combinations of a subset of genes. We call them gene groups. And there are 18 gene groups, which are the row clusters shown here. So this together gives an overall picture across the 33 cancer types. And we also can align the IHAS structure with the pan-cancer sample groups. And those gene groups, 18 gene groups, can be put together into 3 metagene groups. And turns out the combinatorial expressions of those 3 metagene groups can explain many pan-cancer phenotypes. For instance, the mesenchymal subtypes or mesenchymal samples typically have high expressions in metagene group 1 and 2, which are enriched with the immune responses and development genes. And while hypermutated samples typically have high expressions in metagene group 3, which are enriched with the cell cycle related genes. And we can also check that what kind of pathways impact which subtypes of the gene groups or the supermodule groups, and also identify the recurrent hop factors in those supermodule groups. And turns out there are 2 types of pathways which impact a different subset of those metagene groups. But you'll come to my poster to know more about that. And we validate the IHAS structures in over 300 external data sets. So this is a list of them. And I'll just give a few examples. So here we extracted the Metabrick and Rimbrain data from the breast cancer and the GBM, and find out that their combinatorial expression patterns are very close to the patterns we derive from IHAS in TCGA. And we also validate IHAS using the CCLE data, the cancer cell line encyclopedia data, and find out that the associations with the mutations and DNA methylations are relatively well preserved in CCLE, while those associated with the microRNA expressions and the protein phosphorylations are not. And finally, we use the Achilles data of the gene dependency to validate the knockdown effects. Remember that earlier I told you there are two types of pathways which impact different metagene groups. And we can make predictions about their perturbation responses in those metagene groups. And we can use the Achilles data to check their predictions, validate their predictions. And turns out that they're very well compatible with our predictions. So this is it. If you are more interested, just come to my poster. Thank you very much.